Today, what we're going to be discussing in this meeting, we're going to first complete our discussion of the special senses. If you remember last class, we have discussed the vision and the anatomical structure of the eye. Another thing that we started our discussion off was hearing and the anatomy of the ear. Today we are discussing two other sensations that will be detected by the ear as well. Those are going to be two different sensations of the balance. All right, so after we complete our discussion of the special senses, we're going to then discuss more about the autonomic nervous system, which is going to be chapter 13. And after break, we're going to be discussing the endocrine system, which will be chapter 16. So once we complete those chapters, this is going to be everything that you're going to need to know for the last lecture exam. If you remember, our last lecture exam 4, is going to be covering chapters 13, 15, and 16. If we got time at the end, we're going to be going over the most important diagrams that you should be preparing for the last lab exam 4, which covers the nervous system and special senses. So again, today we're having not 14, 15, 16, it's 13, 15, and 16, I believe, or will it be autonomic? Let me double check. You might be right, I don't remember. Yeah, so it's going to be 14, I'm sorry, 14, 15, and 16. Yes, 14, 15, and 16. So both exams going to be available for you by the end of the day today. So expect it to be available for you to complete by midnight today. So it's going to be available for 1 week. It's going to close next Tuesday. It's going to be available for you to complete for 7 days. For those who are asking about the final review, this will not be considered anymore. For the final review, which is worth 40 points towards your grade, it won't be considered. So your final grade for this course is going to be out of 960 rather than 1000. And your grade is going to be a percent out of 960. All right, so I will not be collecting the final review handout. Some of you were asking about the final review handout. I will not be collecting final review handout. We won't have enough time to get this complete. Any general questions concerning the lab exam 4? where to study for the lab exam for. On Canvas, you will have a lab exam for final review that has the most important diagrams that you're going to be tested on. This includes the central nervous system and the special senses. At the end today, at the end of our meeting today, in the last 30 minutes, I will go over the main diagrams, which we have recently covered, which includes the brain, includes the eye, and includes the ear, special senses, and the central nervous system. This is lecture exam 4. This is our last lecture exam. You still have lecture exam 3 open, so if you didn't complete it, get it completed. But we have already covered those chapters tested in lecture exam 3. All right, so what did we mention so far in terms of anatomical structure? And so Nico here, just to make sure I understand, is a review the big document of homework that we did. No, this is a difference. This is the in class. So what you did complete throughout the different meetings, either in class or virtually, those are called the in class activities. And those are going to be curved. So for example, you did complete 8 activities, somebody else did complete 12, somebody else did complete 7. You add all the activities that you have completed into one PDF file that you're going to be uploading to Canvas. And I will look at the average, how many assignments were completed by the class, and your grade is going to be determined according to the average. If you are completing much more than the average, this means you're going to be adding extra credit points. Yes, you need to include the ones that we've completed in class before we stop having our face-to-face meetings. Yes. You can simply scan them or get pictures of them and add them to this file that you're going to be submitting. Last class I said 7 as an example of the average. So I need to collect all the assignment from everybody to determine what's going to be the average, and your grade will be weighted according to the average of your class. Is this clear, Era? The extra credits, the 5 extra credit, those the extra credits that will be when you complete the in-class activities. If you did complete more extra credit, more in-class activities than the average of your class, this is going to be considered your extra credit points. The 5 extra credit points that I mentioned also last time, those for the completion of the instructional evaluation. And you can access this from my FSCJ. Don't wait until the last minute to get this completed, because it usually closes like a week before class ends or something like this. or a few days before the class ends. So get this completed. And once you complete, it tells you, for example, you rate 4, 5, 3, 2, 1 for each of the things that you're going to be rating for the class. And once you get this completed, it will show you a thank you page at the end. Please get a screenshot of the thank you page that will be appearing after you complete your instructional evaluation. And this screenshot, you will be adding it with the documents that you're going to be uploading of the in-class activities. So one of the things that you might be uploading for 5 extra credit points towards your grade is going to be the thank you page, a screenshot of the thank you page that will appear after you complete the instructional evaluation. Five extra points, not 5%. So it's not 5% of the grade. All right. It's on my FSCJ. My FSCJ. So when you open your courses, you will be able to, the courses that you are enrolled to or you are enrolled in, you will see a tab next to the course name. This is where you can have access to the instructional evaluation. So if you did complete this earlier, you can just take a screenshot of the course name that doesn't show the tab present. It's not showing that the tabs that says complete the evaluation. And if it's not showing this tab, this simply means that you did already complete. So take a screenshot of the course name showing that you don't have this tab anymore. So this means that you have already completed it. And this screenshot again will be added to your in-class activities file. All right, you're gonna add the screenshot to your in-class activities file. This is how you're gonna be submitting this. Is this clear for everybody? Any further questions? Any further questions? Concerning exams, concerning when they will be available, how the grading is gonna look like. So what you're gonna need to do right now, check that you've completed lecture exam 1, lecture exam 2, lecture exam 3, and lecture exam 4 will be available for you to complete this week. So by the end of this week, you need to have completed all of the 4 lecture exams. If you didn't complete any of them, please email me. If you didn't complete any of the lecture exams, please email me. Also, you need to have completed lab exam 1, which covers the tissues, lab exam 2, which covers the bones. You should also have completed lab exam 3, which covers the muscles. And lab exam 4 is going to be available for you to complete this week. So we have completed already 3 lecture exams, 3 lab exams, and we still have 1 lecture exam, 1 lab exam. Those 2 are still remaining. So what else should I worry about? If I've already completed 3 lecture exams, and this is the case for most of you. What else should I worry about is in-class activities. And the lab assignments, which are conducted through visible body courseware. You need to make sure that you've completed all the 4 lab assignments. You've got 4 lab assignments that you should have completed so far. Yeah, lab exam 4 is going to be through Canvas, like the bones lab exam, like the muscles lab exam. It's going to be the same way like what we did for lab exam 4. In class activities, again, you're going to be combining all the activities that you have completed in class and virtually when we moved online. Combine them into a single PDF file that you're going to be uploading on Canvas, where it says in class activities and pop quizzes. Upload this single file. You will see a tab where you can upload this file, single PDF file that's going to be reviewed. And again, the grade here, your grade is going to be determined according to the rest of the class performance. So your grade is going to be, the maximum point is going to be the average. All right, so if the class, the average, the average number for completion of the in-class activities was 7 assignments. So for those who did complete the 7 assignments, they will get the full grade for the in-class activities. If you've completed more than 7 assignments of the in-class activities, those extra assignments that you completed beyond average, those are going to be considered extra credit points. All right, so our last in-class activity is going to be conducted today. So from today, you should be able to upload the in-class activities. Starting from today, you can upload your in-class activities. Today is going to be the last in-class activities that will be given to you. All right, so again, what you should worry about, what you should have completed so far. You should have completed 3 lecture exams, 3 lab exams, and you should be aware that you need to complete both lecture exam 4 and lab exam 4. You should also make sure that you combine all the in-class activities that you have completed. And your in-class activities will be weighted according to the average of assignments completed by your class. You need also to make sure that you have completed the 4 lab assignments that you should have completed through visible body courseware. And this is actually a very important thing for you to prepare for the lab exam. You need also to complete the nervous system lab assignments. This will help you studying for lab exam 4. All right, for those who want to complete the final exam. Please email me your class day and time, and that you want to complete the final exam, and I will be sending specific information for you. Final exam replaces the lowest lab exam grade. If you miss the lecture exam, let me know. I know I will open it up for you. The final lab, the final exam, the optional final exam again is optional and is going to be replacing the lowest lab exam. Please email me first name, last name, your class day and time. So for example, I'm attending Tuesday, Thursday at 9 o'clock. So please put TR 900. I'm attending Wednesday, 10 o'clock. So 10 a.m. So Wednesday, 10 a.m. This is what I need to know. And I will be telling you specific information. I will be creating a messaging group on Canvas, and I will be addressing those people who want to take the final exam. I don't want to give the information to everybody for me to not create more confusion. So only for those who are willing to complete the final exam, only for those who need to complete the final exam, they will need to contact me directly. For the part 2 of Tuesday, it was not recorded. It was not recorded. But the information that I did cover on Tuesday was reviewed at the beginning of the Thursday meeting. All right? Hershey? Hershey? Does this answer your question? All right, so the information that I've covered in the second half of the Tuesday class was recorded, but it didn't record. Zoom did not record the sound. So what I did is uploading the first half that has a sound for, and for the second half was not uploaded because it won't make any sense. It doesn't have any sound for it. And the materials that I have covered in the second half of this meeting was reviewed at the beginning of the Thursday class. All right, any questions? Any questions? So your main concerns so far for the next week, which is our last week, complete lecture exam 4, complete lab exam 4, which is going to be available until Tuesday next week. Put together all the in-class activities, and if you want to complete the instructional evaluations, this is going to be for 5 extra credit points towards your in-class activities grade. So you're going to be adding the thank you, a screenshot of the thank you page to your in-class activities files that you're going to be uploading, and this will be giving you 5 extra credit points. Make sure some of the people forget the lab assignments. Some of the people forget the lab assignments. Please make sure you have completed your 4 lab assignments: tissues, bones, muscles, and nervous tissue and nervous system. We have 4 lab assignments that are gonna be completed through Visible Body courseware. Again, if you want to take the final exam, If you want to complete the final exam to substitute a missed lab exam or a layup exam that you haven't scored well on, you need to email me that you want to complete your final exam. You need to include in the email your first name, last name, your class day and time. Tuesday, Thursday, 9 o'clock, Wednesday, 10 o'clock, whatever your class day and time. All right. Any other concerns concerning the lab exams, lecture exams, anything? All right, so what did we discuss so far in the ear? If you remember, we did have three main parts of the ear. We have the external ear and the outer cartilaginous part covered by skin. This is called the ear pinna or the auricle. The auricle function was to capture and collect the sound waves and direct those sound waves through the external auditory or acoustic meatus. And remember, towards the end of the auditory meatus, we did have the tympanic membrane. And remember, this is your eardrum that's going to be responding, vibrating according to the sound waves it will be getting. Those vibrations going to be transmitted from your external ear through the eardrum to your middle ear. And if you remember, in order for me to allow the free movement of the eardrum, I need to equalize the pressure on both sides of the tympanic membrane. So I'm connecting my middle ear to the pharynx by this tube, which is called my pharyngeal tympanic tube. Inside the pharyngotympanic, inside the middle ear, I'm sorry, you've got three tiny bones, three ossicles that will be conducting those sound vibrations through your middle ear. Those are the malleus, incus, and stapes. This vibration is going to be conducted through the oval window to the fluid located within the cochlea. Remember, the cochlea here is the organ responsible for hearing in your internal ear. We have three organs located in the internal ear. The organ responsible for hearing is the cochlea. If I have a cut section of the cochlea, how does it look like from inside? We did see that the cochlea is a bony cavity, and inside this bony cavity, I have a membranous tube. So when we look at this section of the cochlea, we will figure out that the cochlea is divided into three main spaces. This space is located outside of the membranous tube. Those two spaces are located outside of the membranous tube. So we call the fluid inside the bony cavity, but outside of the membranous tube. This is called my exolymph. Compared to the fluid located inside this membranous tube on here, this is called my endolymph. If I'm looking from a side view, this is how the cochlea gonna look like. This is my bony cavity, and traveling in the middle of it, we've got the membranous tube like this. In order for you to hear a sound, you see, this is my foramen ovale that we've seen on here, or the oval window, I'm sorry, the oval window. This is what allows the vibrations to be conducted from the ossicles to the fluid located in here. So now the vibrations are conducted from the bone to the fluid. What do we call the fluid? Again, inside the cavity, but outside of the membranous tubes. This is my axilla. In order for me to hear the sound waves need to have a certain amplitude and a certain frequency. If I am exposed to a very high frequency, I won't be able to detect the sound. If I am exposed to a very low frequency, I still won't be able to detect the sound. Why? If you are exposed to a very high frequency of the sound, very, very high, it will be passing from this chamber, this is my scala vestibuli, to the scala tympani, passing through the scala media, but even before I get closer to where I keep my hearing receptor. So if this is my hearing receptor on here, the sound waves, if I'm detecting a very high frequency sound, it will pass unnoticed. Why? Because it will be penetrating the scala media, this chamber in the middle here, this is what we call scala media. It will penetrate even before I reach the receptor responsible to detect hearing, which is the organ of Corti. What if you have a very slow frequency, very low frequency of a sound, like this? This frequency of the sound will fail to penetrate through the scala media, it will fail to pass through this membranous tube. And if it failed to travel through the membranous tube, I won't hear the sound. So if I have a very high frequency or a very low frequency of a sound, I still can't hear those frequencies. The frequencies that can penetrate the scala media, causing the activation of the organ of Corti, are gonna be between 20 to 20,000 hertz, or 20 to 20,000 hertz. This is the vibrations I can hear. If I have a much greater frequency of the sound, it won't work. If I have sound which have lower frequency than 20 hertz, I won't be able to penetrate through the scala media. Again, why do I need to penetrate through the scala media? Why do I need to travel from the scala vestibuli to the scala tympani, passing through the scala media? Because this is where I have the receptor responsible for hearing, the organ of Corti. The organ of Corti is going to be formed of hair cells with cilia, and those hair cells did have a gelatinous membrane on top of the cilia. This is what we've called, if you remember, the tectorial membrane. Tectorial membrane, it's the gelatinous membrane that will be attached to the cilia of the hair cells. So if I was capable to allow those vibrations to pass from the scala vestibuli to the scala tympani, traveling through the scala media means I'm causing the vibration of the endolymph of the scala media. And this will cause the movement of the tectorial membrane, causing, if I'm magnifying here just one hair cell of the organ of Corti, this will cause the movement of the cilia of the hair cells, causing the opening of sodium channels and allowing those cells to depolarize. Remember, when I open sodium channels, this will allow sodium in. And once you allow sodium in, you create a state of depolarization. And this depolarization of the cell gonna be a signal. If you get enough stimulation of the organ of Corti cells, you're gonna be able to create an electrical impulse, an action potential that has the ability to travel for long distances, traveling through this nerve. We call the nerve that carries the signals responsible for hearing. It's the cochlear division of the vestibulocochlear nerve. So how can I hear again? The organ of Corti located in the scala media of my cochlea will become activated, and once it becomes activated, it will be forming electrical impulses that will be traveling up to your medulla, then to your midbrain, then to the thalamus, and then from the thalamus it gets distributed to your cerebral cortex for you to be able to detect this sound. For me to be able to hear, I need a sensory area in my cerebral cortex, if you remember. So the function of the pharyngotympanic tube, what was the function of this tube that will be connecting your pharynx to the middle ear? Pharyngotympanic tube. Why do I have a connection between my middle ear, which is a bone cavity inside my temporal bone, to my pharynx, to allow the air to flow in whenever there is a change in the atmospheric pressure, for me to equalize the pressure on both sides of the tympanic membrane? Exactly. So here, what is my function? It's to equalize the pressure on both sides of the tympanic membrane. Moving on to two other organs that will be located in the internal ear. Those are the vestibule and the three semicircular canals. So again, how many organs do I have in my internal ear? Got three organs. First one was responsible for hearing, and this is going to be my cochlea. Another organ here is going to be my vestibule. And the third organ is going to be the three semicircular canals. The vestibule and the semicircular canals are going to be responsible to detect equilibrium and orientation. The vestibular receptors will be monitoring the static equilibrium. Compared to the semicircular canals, they will be monitoring the dynamic equilibrium. What's the difference between static equilibrium and dynamic equilibrium? Static equilibrium is going to be your ability to detect the position of your head in space. And this is going to be allowed by the vestibule. The vestibule has two compartments. It's a bony chamber. It's divided into two smaller parts, the saccule and the utricle. Saccule is going to be continuous with the cochlear duct, and the utricle is going to be continuous with the semicircular canals. Inside the utricle and the saccule, we're going to have the receptor responsible to detect the static equilibrium. This is going to be my maculae. So I have two maculae, one located in the utricle and one located in the saccule. If you see on here, this is the utricle, this is the saccule, and inside of each of them, you're gonna have this receptor. Again, what do we call the receptor responsible for static equilibrium? It's the macula. So what does it mean to be able to determine the position of my head in space? So if you bend your head forward, when you close your eyes, you don't see anything, you bend your head forward, would you be able to know that your head did bend forward? Or you bend your head to the right, or you bend your head to the left while you're closing your eyes? Would you be able that your head has changed its position in space? What do you think? Yes. How are you able to? Inside the vestibule, inside the saccule and utricle of your vestibule, you have hair cells similar to the ones that we've just seen in the organ of Corti. But those hair cells are slightly different. They have stereocilia and kinocilium. I have two different types of cilia in those hair cells of the macula. I have kinocilium and stereocilium. So when the kinocilium, those ones on here that have the ability to move, will be bending towards the kinocilium, the stereocilium, I'm sorry, on here. Stereocilia are bending towards the kinocilium. This will open sodium channels. So what's going to be the state of this cell is going to be, if you open sodium channels, what's going to happen to this cell? How activated? So we have two electrical states, remember? When you open sodium channels, what's my electrical state? Inside will become more positively charged, outside will become more negatively charged. What do we call this state? Depolarized. If the stereocilia are moving away from the kinocilium here, this will cause the opening of potassium channels. And remember, when you open potassium channels, what's going to happen to the cell membrane? Potassium is going to be leaving the cells, making the inside to be even more negatively charged than the resting membrane potential. We call this as a hyperpolarization. So how do you know what is the position of the head in space, of your head in space? You have thousands and thousands of those hair cells, and you're going to have stereocelia and kinocerium in all directions. So try to imagine you have only two hair cells, one in your right ear and one in your left. And each of them has only one stereocelia and one kinocerium, like this. This is not the situation. It's much more complicated in real life. This is just a simplified way to look at, just for you to imagine how your brain is able to figure out the position of your head in space. So if you are bending your head to the right, this is my head on here, and I'm bending my head to the right. So what's going to happen? This stereocerium is going to be moving away or towards the kinocerium. Towards or away from the kinocerium. Away. How about on the other side, in my left ear? The stereocelia is going to be moving away or towards the kinocerium? Towards the kinocerium. Remember, if I am moving away from the kinocerium, this is going to be opening potassium channels. So potassium is going to be moving out. So what is going to be the state of this cell? It's going to be hyperpolarized. How about in this cell? If I'm moving towards the kinocerium, what's going to happen? I will open sodium channels. So sodium is going to be moving in. And when I move in, what's going to happen to this cell? It's going to be more positive on the inside compared to the outside. This means I am depolarized. So I'm sending a depolarization signal from my left ear, and my right ear is going to be hyperpolarized. My brain will get those two signals and will be figuring out according to the state of hyperpolarization on the right side and the state of depolarization of the left on the left side. This means that my head is bent towards the right side. This is a very simplified way to look at how your brain can figure out the position of your head in space. It's much more complicated than this, because you're going to have stereocelia and kinocerium all around, and your brain is getting codes of zeros and ones. Zero means I am hyperpolarized, ones means I am depolarized. And he will be decoding all those signals coming from, again, what did we call the name of the receptor responsible for static equilibrium? My macula. And what was the organ where those two maculae that we have are going to be located? What organ is it? Remember, we did have three organs in the internal ear. Cochlea, which is responsible for hearing. What was the other organ responsible to detect static equilibrium having maculae inside. This is my vestibule, exactly. Vestibule. So the organ is the vestibule, and this is something, as this is one of the most common wrong answers I usually see. I ask you about an organ, and your answer is going to be a receptor. I ask about the receptor, and your answer is going to be an organ, and so on. So the organ is the vestibule. The receptor inside the organ responsible to detect static equilibrium is the macula. And how many of them do we have? We've got two. One is the utricle, one is the sacral. If we're looking closer to the structure of the macula, you will see here hair cells, stereocilia, kinocilium. But those cilia coming out from the hair cells, they are embedded in an autolytic membrane. A membrane that is going to be formed of calcium stones. Why? You don't want to have any external effect on those cilia. You don't want to have any vibrations causing the movement of those cilia. Try to imagine that any vibrations that you didn't have this autolytic membrane, you don't have this calcium stone membrane covering those cilia. What's going to happen? Any kind of vibrations that you get exposed to, you won't be able to figure out the actual position of your head in the space. Can you imagine? So in order to prevent something like this from taking place, what will I do? I will be embedding those cilia inside a calcified membrane. Does this make sense? Questions? Questions? All right. Moving on to our third and last organ of the internal ear. This is going to be the semicircular canals. We have three semicircular canals: anterior, lateral, and posterior. Those are the same like the cochlea. They are cavities inside the bone, and inside which you're going to have the membranous ducts. So you have a cavity, membrane, bony cavity, and inside those bony cavity, you're going to have the semicircular ducts. At the base of each of those ducts, we're going to have a bulging part. We call this is the ampulla. If this is a semicircular canal like this, towards the lower end, you're going to have this bulging region. We call this is the ampulla. And this is the site where you're going to have the receptor responsible to detect the dynamic equilibrium. So again, what was static equilibrium? This was your ability to determine the position of your head in space. Dynamic equilibrium is about the angular or rotational movements. So let's have a look at the inside of the semicircular canals. At the base of each of them, you see on here this bulging part. This is called the ampulla. And this is where I have the receptor, and the receptor responsible to detect the dynamic equilibrium is what we call the crista ampullaris. Crista ampullaris, this is the receptor responsible to detect the dynamic equilibrium. Are you able to detect the movement? What do you think? Would you know that you're moving forward, backward? What do you think? Yes? Are you able to detect motion, movement? So if you're moving forward, backwards, are you able to detect those movements? Are you moving right now along with all your house? If you're sitting still, are you actually moving right now? If you are sitting still, you're not moving any body part of yours. Is your body is moving? Yes, definitely. With the buildings, with the street, with everything is gonna be moving. Why? Because exactly in the Jeep, you are gonna be part of the Earth, which rotates around its own axis and rotates as well around the sun, right? So are you currently moving? Are you currently rotating? Yes, you are moving. Are you able to detect this movement? No, because you don't have the ability to detect movement. What do you have is your ability to detect the change in speed. So if you are in your car, you're driving, do you feel that you are all the time pulled forward, all the time, all the trip? When would you feel that you're moving, especially on a train? When you are accelerating, you are increasing the speed, or you are decelerating when you're reducing the speed. So what I feel is not the movement, I feel the change in the speed of the movement. Is this clear? Is this concept clear? This is what we call again the dynamic equilibrium. My ability to know whether I'm speeding up or slowing down. Why do you think you're gonna have three semicircular canals? Why I don't just have one or two or 10 or unlimited number of semicircular canals? Why would I have three semicircular canals on here? Yes, there is three dimensions that you can move in. So you can move forward, can move backward, can move up, down. I can move from side to side. Those are the three dimensions that you can move along. So you need three semicircular canals to cover those three dimensions that you can move within. All right, so how do I detect that there is a change in the movement? So try to imagine you have a piece of paper like this, and you have a marker, cylindrical pencil or marker, whatever. And I'm pulling this sheet of paper. So when I pull, this means that I did increase or reduce the speed of this sheet. I did here when I pull it, I did increase or reduce the speed What do you think? So the sheet of paper on here is not moving, and you decided to pull it in this direction. Are you increasing the speed or reducing the speed of this paper? Do you understand my question? What do you think? You are starting from 0 and you are moving, so you are increasing the speed, right? So where this marker is going to be moving, is it going to be moving in the same direction as the sheet or in the opposing direction? What do you think? If I have a marker on the sheet and I'm pulling this sheet, will this marker move in the same or the opposite direction? So just put a piece of paper in front of you, put a marker, and just pull it, increasing the speed. You will see that the marker is rolling in the same or opposite. What do you think? Let me ask you the question in a different way. You are in a car and you are speeding up. Speeding up means what? Means that the car is moving, let's say from 30 miles per hour to 90 miles per hour. So where your body will go when you speed up, would your body move backwards or forwards? Backwards. So if I am moving at a faster speed, I'm speeding up, there is an acceleration. What's going to happen to the content inside? My body is going to be moving in the opposite direction to the movement. So the car is moving forward, my body is going to be moving backwards. What if this person here while he's speeding, he hits a tree? So what's going to happen? I was moving at 90 miles per hour. Now I stopped. Now I became 0 miles per hour. So where my body will be moving? Will it be moving backwards or forwards? Forwards. So now, if I am decelerating, I'm reducing the speed. The car was still moving in the same direction. There is no change in the car direction. The car is still moving forward. But my body responds differently according to whether the car is accelerating, is speeding up. If I'm accelerating, what happens to my body? My body is moving backwards in the opposite direction. What happens in here? When I decelerate, when I reduce the speed, my body is going to be moving in the same direction as the car. All right? So in both conditions, the car was moving forward. If I am accelerating, my body is moving in the opposite direction. If the car is decelerating, my body is now moving in the same direction. Does this make sense? All right, so let's apply this on your semicircular canals. Again, inside at the base of each of the semicircular canals, we're gonna have a crest and elevation like this. And on top of it, you're gonna have the hair cells, like the ones that we've seen in the cochlea, like the ones that we've seen in the macula. And on top of the cilia of the hair cells, you're gonna have a gelatinous cap. We call this is my cupola. If you see on here, this is the cupola. We call the receptor inside the ampulla of the semicircular canals. This is my crista ampullaris. The elevation in the ampulla, crista ampullaris. So what happens to the crista ampullaris if I'm rotating here in this direction? The fluid inside my semicircular canal, where will it go? If this is if those are my hair cells in here, inside my crista ampullaris, those are my hair cells like this. And this is the cilia. Remember, inside the tube, you've got inside the semicircular canals, inside the cochlear duct, inside the utricle and sacule, you got endolymph, you got fluid. So if you are moving from 0 to 10, your speed. So this means I am accelerating, I am moving faster. So what's gonna happen to the fluid inside? I will be drawing the fluid here in blue. Fluid, will it be moving in the same or the opposite direction? What do you think, 1 or 2? If I am speeding up, you're gonna be moving in the opposite direction. So you're gonna be bending the cilia of those hair cells in this direction. What if I am still rotating in this direction, but I'm moving slower? These are my same hair cells. This is still my cupola. Here's the gelatinous cap of the crista ampullaris. And here I'm moving from, rather than 0 to 10, I'm moving from 10 to 0. I am decelerating. When I decelerate, the fluid inside the semicircular canal, will it be moving in the same direction or the opposite direction? 1 or 2? If I'm decelerating, I'm slowing down. Remember, when you slow down, the body did move in the same direction. So again, when I slow down while I'm rotating or I'm moving, it's gonna be moving in the same direction as the rotational movement. And if I am moving in the same direction, this is gonna be bending the cupola in the opposite direction, bending the cilia of the hair cells in the opposite direction. So my brain will be getting signals if I'm accelerating different from when I decelerate. So your brain will tell you now you are activating this semicircular canal. This means that you're moving up or down. But I don't know whether you are accelerating or decelerating. It's according to where did I bend the cupola. So where am I moving is going to be determined by which semicircular canal of the three that you've got will be activating its crista impellars. This is for the direction. Which side? Am I going up, down, towards the right side, towards the left side? Am I going forwards, backwards? This is going to be determined according to where the brain is getting the signals. Am I getting the signals from this semicircular canal, from this semicircular canal, or from this semicircular canal? Each one is going to be covering one dimension that they can move with it. But how my brain will be able to figure out whether I am speeding up or slowing down, it's according to whether you did depolarize or hyperpolarize, according to whether did you bend the scapula in this direction or the opposite direction. And this will take place simultaneously in both ears. And according to the signals sent from the semicircular canals in both ears, my brain will start to decode those signals and will be figuring out what direction and whether you are accelerating or decelerating. What do we call this sensation? My ability to know whether I'm accelerating or decelerating. This is what we called, again, any idea? Dynamic equilibrium. All right, so for the first in-class activity for today, please complete this table on here. All right, please complete this table on here. So those two belong. Dynamic equilibrium, this is one thing. Those again are the organs in the inner or internal ear. Any questions? Any questions? All right, we can have break for 10. We come back, discuss more about the autonomic nervous system and the endocrine system. All right, so welcome back. Just to make sure that you are all aware of the presence of this review handout for lab exam 4. If you go here under the lab activities files on Canvas, you will find the lab exam 4 review PowerPoint, and this is how it's gonna look like. This is what I will be going over at the end of today's class. All right, it's gonna have the brain, the eye, and the ear. All right, those are the most important diagrams that you need to know for the upcoming lab exam. All right, is this clear? Is this clear? Yeah, the lab exam is gonna be available for you starting midnight tonight until midnight on Tuesday, and this will be applied for both lecture exam 4 and lab exam 4. No, you don't need to schedule for the exam. What you will need to schedule for and arrange with me is the final exam, optional final exam. If you decided to take the optional final exam, you need to send me, as I mentioned earlier, your first, last name, class, day, and time. This is only if you are gonna be taking your the optional final exam to substitute one of the 4 lab exams. But for lab exam 4, lecture exam 4, those are gonna be available starting midnight tonight until midnight next next Tuesday, this Tuesday. All right, is this clear? In-class activities, all in-class activities need to be combined into one file that you're going to be uploading on here. See, it's not available for you yet to upload. I will also make it available for you tonight. On here, you will go directly to this in-class activities and pop quiz, and you can upload this files that you've created by combining all the in-class activities, either the ones that you've completed in class as well as the ones that you've completed in our virtual meetings. Either you're attending them live or you have been watching them recorded. All right, you're gonna be uploading here the files that you're gonna be creating. You go in class activities and pop quizzes, and it will show you a place where you can upload your document. All right, is this clear? Is this clear for everybody? So just put all what you've completed. You don't have to put them in any order. And I will look at what you've completed and what you did not and compare again. I will compare it to the average of the number of assignments that have been completed by the rest of class. So your performance is gonna be measured according to the rest of the class. Alright. Right, so today we are starting our discussion here of the endocrine system. If you remember, the endocrine system was one of the two main control centers in your body. The first control centers that we've been discussing was the nervous system. It's going to be controlling the rest of the body organs by sending out electrical signals through the nerve fibers. Here in the endocrine system, I have glands which are going to be releasing hormones, chemical messages through the bloodstream. We classify the glands according to whether they are releasing their secretions directly in the bloodstream or through a duct. So we're going to be classifying the glands. If they are releasing their secretions directly in the bloodstream, we call those are the endocrine or endocrine glands. If they are releasing their secretions, like the salivary gland, for example, the parotid gland, it releases the saliva through a duct. So we call this is an exocrine gland. So again, if I'm releasing secretions directly within the bloodstream, not through a duct, it's an endocrine gland. If the gland is releasing its secretions through a duct, it's an exocrine gland. We classify those hormones, the chemical messages that we're going to be releasing directly in the bloodstream. Here, we're concerned here with the endocrine system. So we're studying the glands that will be releasing hormones in the bloodstream. We classify those hormones according to the chemical structure into two major categories. Those are the amino acid based hormones. They have a protein nature. Those like the amines, thyroxine, the peptides, and proteins. And we've got steroids. Steroids are like the gonadal hormones, adrenocortical hormones. Here, I'm a protein. Here, I am a cholesterol-based hormone. So this is a fat, this is a protein. Is there any difference in terms of action and how they would be activating their target cells? So you are releasing, why do you release the hormone in the first place? For you to conduct a certain action on a target cell. So this is my target cell. The hormone needs to have an effect on this target cell. Like for example, a nerve cell that send out an electrical signal to the muscle fiber for the muscle fiber to contract in response to this signal. All right, so here, hormone is going to be the same concept. I am conducting a certain action on a target cell. How those hormones going to have their actions conducted on the target cells? Some of the hormones can be changing the membrane permeability of their target cells, so allowing ions to travel in or out of the cells, so creating a state of depolarization or hyperpolarization. They might also induce protein synthesis. I'm inducing the protein formation. Like what? Like, for example, you will hear a lot about people taking hormones, steroid hormones, to induce the, the muscle growth, for my muscle to become larger in size. I'm taking external hormones that will be inducing their enlargement. All right, so how did you enlarge those muscles? By creating proteins inside. So it might be hormones that induces the protein synthesis. Also, another function of a neuron by which it's going to be acting on its target cell is to activate and deactivate enzymatic systems inside the cell. So some hormones might be inducing the metabolism to break down sugar, for example, and release energy and heat, like the thyroid hormone. Thyroid hormone is doing what? It enhances your cells to break down the fats, proteins, and sugars to generate heat. I am a calorigenic hormone. So I'm inducing those enzymes that carry on those activities. So another function here of those hormones will be to activate or deactivate certain enzyme systems within the target cell. That might also be inducing the secretory activity of the target cells. They might induce mitotic division, like as we're gonna see, the growth hormone. Growth hormone is allowing you to grow. So how the growth hormone is gonna be acting, it enhances some of its target cells to divide. And as you divide your cells, this is gonna allow you to grow. So what is the difference in terms of action between a protein, an amino acid based hormone, and a steroid hormone? An amino acid based hormone, I am formed of amino acids which contain charges in most of the cases. So I am charged. Can I pass through the phospholipid bilayer of the target cell in order to induce my action? Can I simply pass through the phospholipid bilayer? If you remember, what was the structure of the phospholipid bilayer? It did have a phosphate group directed outside of the cell membrane, and it did have those fatty acid tails. So if I'm charged, am I lipophilic? Am I in love with the fat here, with those fatty acids? Can I travel through the fatty acids? No, I cannot, exactly. I can't pass through the fatty acids. I can't. So what would I need to have an action on my target cell is to bind to a surface receptor. Once the hormone binds to its receptor on the outside surface of the cell membrane, it will allow the activation here of a protein, we call this protein is G protein. Why do we call it G protein? Because simply it gets activated by binding to a GTP, which is very similar to the ATP. So once I get a hormone receptor complex formed, this induces the activation of G protein by breaking down a GTP. This activated G protein will then go and activate an adenylate cyclase. This adenylate cyclase is going to be breaking down an ATP into cyclic AMP, into an adenosine monophosphate. This cyclic AMP is going to be activating specific protein kinases according to the type of the cell it's located inside. And those protein kinases are going to be triggering the response of the target cells by activating enzymes, stimulating cellular secretion, opening ion channels. We call the cyclic AMP on here, it's my second messenger. So who was the first messenger? It was the hormone itself. So again, how an amino acid based hormone gonna have an effect on the target cell? It's gonna need to bind to a surface receptor. And when it binds to a surface receptor, this is gonna cause the activation of a G protein by allowing a GTP to bind to it. Once the GTP binds to the G protein, this is gonna be in activating the G protein, and the activated G protein activates adenylate cyclase that transforms an ATP into cyclic AMP. This cyclic AMP will activate protein kinases, and those active protein kinases gonna be the ones that will induce the response to take place on those target cells. What are examples here of hormones that act through activation of adenylate cyclase and forming cyclic AMP? Those are like the epinephrine, the adrenocorticotropic hormone, the FSH, LH, glucagon, the parathyroid hormone, TSH, and calcitonin. All those are hormones which are protein-based, amino acid-based hormones. How about the steroid hormones? Steroid hormones, those are lipid soluble. I am a cholesterol-based hormone. I have no problem passing through the phospholipid bilayer. So as I travel to the inside of the cell membrane, in order for me to induce the activity, to trigger the response of the target cell, those lipid soluble hormones, the steroid based hormone, need to bind to an intracellular receptor. See on here, this is my intracellular receptor that I will be binding to inside the cytoplasm of the cell. Once I've got the hormone receptor complex, this hormone receptor complex is going to be traveling to the inside of the cell and will be targeting specific genetic codes. And it's going to be turning on those genetic codes that are coding for a specific protein. So let's say I am an androgen. I am a male sex hormone. And this is my target cell. This is a muscle cell. So the androgen will simply be able to travel through the plasma membrane of the cell it's gonna bind to an intracellular receptor, not a surface receptor. Once the androgen binds to its receptor, forming a hormone receptor complex, this complex will be activating specific genetic regions on the DNA. Like for example, it's going to be inducing the formation of myosin and actin filaments within the muscle cell. And this is what causes the muscle cell hypertrophy if a person got extra androgens from outside. And this explains also why males will have a larger muscle bulk compared to females because of the presence of larger amounts of androgen. So according to what the gland will be deciding to release more or less of a specific hormone, we have three main regulatory mechanisms for the hormonal release. First one is going to be the changes that takes place in the chemistry of the blood. So for example, if I have a hormone that gets released in order to reduce the blood sugar level, insulin. So what was the stimulant that induced the insulin release? It was an increase in the blood sugar. After I eat, I have an increase in the blood sugar levels. This will be inducing the release of insulin. So the main stimulant to the hormone release in here is going to be the change in the blood chemistry. We call this as a humoral regulation. The examples that you see on here, we've got 4 tiny glands located in the neck. Those are called the parathyroid glands. And they are responsible to release PTH, parathyroid hormone. PTH function is to increase the blood calcium level. So what is the main triggering factor that will be inducing those glands to release PTH? Will be a decline in the blood calcium levels. So whenever I have a reduction in the blood calcium, this will directly stimulate the parathyroid glands to release PTH, and then the PTH will be increasing the blood calcium levels back to normal. So what triggered this gland to release its hormone is the change in the blood chemistry. We call this regulatory mechanism. It's the humoral regulation. I might be also regulating the hormone release by the nervous system. So part of the nervous system is going to be increasing my heart rate, increasing my blood sugar levels in response to stress. This is called the sympathetic nervous system. Sympathetic nervous system induces its action or part of its action by stimulating a gland located above the kidney called the adrenal gland to release the adrenaline in the bloodstream. So the adrenaline will it be its action? It's going to be increasing the heart rate, increasing the blood pressure, increasing the blood sugar levels. All those are actions due to the adrenaline rush in the bloodstream. And again, what was the triggering factor that induce the adrenal gland to release adrenaline out in the bloodstream? It's the sympathetic nervous system. So you see here, sympathetic nervous system also might induce the release of a certain hormone. We call this regulatory mechanism. It's a neural regulation. I have the nervous system here taking action to regulate the hormonal release. I might also have another gland that releases hormones to regulate the hormonal release from other glands. So I have a hormone that induces the release of another hormone. So we call this as a hormonal regulation. So again, we have three types of hormonal release regulation. We have humoral regulation. This is again determined according to the changes that will be taking place in the blood chemistry. We have neural regulations, in which I have the nervous system inducing the release or inhibiting the release of certain hormones. I have hormonal regulation, in which a gland releasing a hormone to induce the release of another hormone. So the stimulant here is a hormone. We call this as a hormonal regulation. Looking at the different glands on here that we will be discussing in this chapter, if you remember, a small gland pending down from the brain attached to the hypothalamus. This was the pituitary gland. This is the master gland of the endocrine system. It regulates a lot of other endocrine glands. If you're moving down to the anterior aspect of the neck, we've got a butterfly-shaped gland. This is called the thyroid gland. And on the dorsal surface of this gland, we're going to have four tiny glands that are located close to the thyroid. So we're going to call them the parathyroid glands. Para means close to or nearby. So four tiny little glands located close to the thyroid glands. Those are my parathyroid glands. We have another gland that will be located in the chest. It disappears as a person grows. It's going to be much more prominent at younger ages. It's called the thymus gland. Located above the kidney, we've got pyramidal shaped glands. Remember, something related to the kidney is called renal. So when you are having a gland located above the kidney, we're going to call this gland the adrenal gland on top of the kidney. Another name for the something related to the adrenal, remember, something also related to the kidney might be called nephro. So what do we call the doctor that treats kidney diseases? Nephrologist. So nephro is kidney. So above the kidney is epinephro. Epi above, nephro kidney, like adrenal above the kidney. So we're gonna see the other name of the adrenaline is called epinephrine. Epi above, nephro kidney. Epinephrine is the same as the adrenaline. Another gland that we're going to be studying in this chapter will be the pancreas. which is located in the abdominal cavity. The gonads also going to be responsible to release the sex hormones, and those are the ovaries in females and the testes in males. So again, what are the different glands that we are looking at in this chapter? First glands that we're going to be studying in details in this chapter will be the pituitary gland. Next is going to be the thyroid. Next is going to be the parathyroid. We won't be discussing much about the thymus gland. We're going to have just a brief introduction about it. We're then going to discuss the different parts of the adrenal glands and the hormones released from the adrenal glands. Finally, we're going to be discussing in details the hormones and the hormonal effects of the sex hormones released from the ovaries in females and the testes in males. Questions? Anybody still awake? All right, so starting first with the pituitary gland. The pituitary gland is, if you remember, is pending down from the hypothalamus. If you remember, what did we give a nickname? What was the nickname that we've given to the hypothalamus when we studied the central nervous system? Not the vice president. The vice president was the thalamus itself. Hypothalamus was secretary of the office, right? Do you remember? So hypothalamus was regulating hormonal release, regulating your blood pressure, regulating your body temperature. Those are the basic activities. The vice president, the thalamus above it, this one was responsible to sort, edit, and relay the information going to the cerebral cortex. If you remember here, pending down from the hypothalamus, we've got the pituitary gland. Pituitary gland is going to be divided into two main parts. We've got the anterior pituitary gland and the posterior pituitary gland. Anterior pituitary gland, as you see on here, it's going to be formed of glandular cells. Those glandular cells are going to be regulated by the nerve cells from the hypothalamus. Compared to the posterior pituitary gland, it's just a pouch where you're going to be storing the hormones released from the hypothalamic nerve cells. So what is the structure of the anterior pituitary gland? I'm formed of glandular cells. I have an adenohypophysis. What I mean by adenohypophysis, it's a gland. I am formed of glandular tissue. Compared to the posterior pituitary gland, it's not formed of glandular tissue. It just serves as a pouch where you're going to be storing the hormones directly released from the hypothalamus. So we're going to call the posterior pituitary gland, which is formed of nerve cells, it's my neurohypophysis. What are the hormones that will be released first from the anterior pituitary gland? The first hormone is going to be the growth hormone. From its name, What do you think is going to be the action of the growth hormone? Mitosis, and generally speaking, it's inducing cellular and organ growth. So what are the target organs for the growth hormone? What do you think is the growth hormone is going to be acting on? What specific cells in your body would the growth hormone act on? It acts on all the body cells, more and more action on your bones and muscles. So what if I have a hyper secretion of the growth hormone? I will have gigantism. I have a tall stature, long bones. What if I have hyposecretion of the growth hormone? I would have dwarfism. So again, the first hormones that we're looking at in here released from the anterior pituitary gland is going to be the growth hormone that induces the bone and skeletal muscle growth. Also, it encourages the cells to use the fat as a source of energy. Another hormones that we're going to be releasing from the anterior pituitary gland, it's the hormones that will stimulate the thyroid gland. We call this as the thyroid stimulating hormone. So what's going to be the action of a hormone called thyroid stimulating hormone? It's going to be stimulating the thyroid gland to release the thyroid hormone. Another hormone that gets released from the anterior pituitary gland is going to be the adrenocorticotropic hormone, or ACTH. What do you understand just from the name of the hormone? It's a hormone that trophic, or tropic means it induces or activates cortico of the adrenal. It induces the release of the hormones from the adrenal cortex. So the adrenal gland, if you remember, is this pyramidal shaped gland located above the kidney, is going to be divided into two regions. An outer part, this is called my cortex. And an inner part, this is going to be my medulla. So the ACTH is going to be inducing which part? It's going to be inducing the cortex of the adrenal gland. So that's why we call it adrenocorticotropic hormone, ACTH. Other hormones that will be released from the anterior pituitary gland, and those are what we call the gonadotropins. What are the gonads? Gonads are the testes in males and the ovaries in females. And what I mean by tropin is to induce. So those hormones are going to be inducing the gonads. Those are two major hormones, the gonadotropins. We have the FSH, follicle stimulating hormone, and the LH, the luteinizing hormone. So FSH and LH are gonna be both released in both males and females. Generally speaking, the action of FSH is to induce the gonads, the testes in males and the ovaries in females, to form the gametes in males to produce the sperms, and in females to produce the eggs. Compared to the luteinizing hormone, it also acts to activate the gonads, testes, and ovaries, but this time is not mainly to induce the formation of the sex cells, the sperms and eggs. This time is to induce them to release the sex hormones. For males, those going to be the androgens, like the testosterone, and in females, the female sex hormones are like the estrogen and the progesterone. All right, so again, two gonadotropins that we've got released from the anterior pituitary glands. Those are FSH, LH. FSH, follicle stimulating hormone, is going to be inducing the production of the eggs and sperms. Luteinizing hormone, this is going to be inducing the gonads to produce the sex hormones, testosterone and estrogen and progesterone in females. Another hormone that gets produced from the anterior pituitary gland, it's the prolactin. Prolactin, from its name, what can you understand from the name here, prolactin? Pro means pro, it precedes. And lactin, here, what do I mean? What is lactation? Lactation means milk production, right? Breastfeeding. So this is a hormone that's gonna be preceding lactation. We call it prolactin. So when do you think you're gonna have a rise in the prolactin levels? Yeah, late pregnancy, exactly. During late pregnancy, I'm getting ready to induce the milk production in the breast because I'm about to have a baby, so I need him to get fed. So I need to induce the milk production in the breast, and that's why you're gonna have a rise in the prolactin levels during late pregnancy, during the end of pregnancy. What's gonna be stimulating further prolactin secretion is the suckling effect of the nipple. And the nipple, when it's sucked, this will induce the hypothalamus to release a hormone or a neurotransmitter, because I'm getting this released out from a nerve cell, but actually we're going to consider it a hormone because it's still going to be circulating in the bloodstream. This hormone that gets released from the hypothalamus is called the prolactin releasing hormone. So this is a hormone that will be inducing the anterior pituitary gland cells to release the prolactin. And again, prolactin is responsible for production of milk. Polycystic ovary is this what you're asking? So in a polycystic ovary, I have follicles that failed to rupture to release the egg. So I don't have a negative inhibition. So whenever I have a follicle rupture in the ovary, if this is the ovary on here, once the follicle ruptures, it will form something called the corpus luteum. And the corpus luteum is going to be releasing progesterone. That will be inhibiting the hypothalamus and anterior pituitary gland from releasing FSH and LH. But one of the hormones also that might get affected is going to be the prolactin. So if I don't have this inhibition, I'm mainly the main function here of the progesterone that gets released when the follicle gets ruptured and ovulation takes place is to inhibit those anterior pituitary gland from releasing those two hormones, FSH and LH. But also, I will be partially inhibiting the release of the prolactin releasing hormone that induces the formation of prolactin. So it will be slightly increased. But a major reason of a person having high prolactin levels in his blood is going to be a pituitary gland tumor, where those cells are getting rapidly proliferating and they are releasing larger than normal amounts of prolactin. All right? So yes, it would be partially increased, but it's not the thing that will be the main concern of the polycystic ovary syndrome. All right? Does this answer your question, Rachel? It's not going to be the main concern. The main concern is going to be disturbed menses. So the menstrual cycle, it's not regular. Patients is going to have infertility. I'm not able to get a child. I'm not sure why. What is the reason behind it? Those what will be the concerns that will get this patient to go to a gynecologist. All right, so again, the suckling of the nipple itself is going to be inducing the hypothalamus to release prolactin releasing hormones that stimulates the glandular cells in the anterior pituitary gland to produce prolactin. And again, the prolactin effect will be to stimulate the mammary glands to produce the milk. I'm engorging them, I am filling them with milk to prepare the breast for lactation. I'm gorging and filling the glands, the mammary glands, with milk. And again, this takes place during late pregnancy. Moving on to the posterior pituitary gland hormones. We've mentioned few hormones, and all those hormones are again produced from the anterior pituitary gland. We've mentioned the growth hormone, TSH, thyroid stimulating hormone, ACTH, adrenocorticotropic hormone, and gonadotropins. Those are like FSH and LH. Another hormone that we've mentioned that gets released from the anterior pituitary gland is going to be the prolactin that induces the milk production in the breast. Moving on to the posterior pituitary gland. Posterior pituitary gland is going to be releasing two hormones. Again, those hormones are not going to be produced by glandular cells within the pituitary gland. Rather, they will be produced from the nerve cells moving down from the hypothalamus. The two main hormones that will be produced from the posterior pituitary gland, those are the oxytocin and ADH. If you remember in our very first class meeting, we've mentioned oxytocin to be a hormone that induces the uterine contractions during labor. And we mentioned it as an example of a positive feedback. So one of the main functions of oxytocin is to induce the uterine contractions, as we've stated back in chapter 1. Remember, the stretch of the uterus will induce its contraction, and the contraction of the uterus gonna be stimulating the hypothalamus to release oxytocin that when delivered to the uterus, it induces more uterine contractions to take place, and as a result, this allows labor to take place. So the baby gets out by increasing the contractions of the uterus. Another function of the oxytocin will be to induce the milk ejection by contracting those lactiferous sinuses. If you see on here, if we're looking at the breast, the breast is formed of mammary glands surrounded by fat tissue. The mammary glands are going to be stimulated by the prolactin to form the milk. This milk is going to be stored in those spaces. We call those are the lactiferous sinuses. So female during a lactation period, she will not be dropping the milk all the time from her breast. This milk will be stored in sinuses until when? Until the baby starts to suckle the nipple. This what induces the hypothalamus to release the oxytocin. Once the oxytocin levels has been increased, this will induce the contraction of the smooth muscles of the lactiferous sinuses on here, and as they contract, they will be ejecting the milk out. We call this is milk let down or milk ejection. So for normal lactation, what are the two hormones that we need to have? For normal lactation, what are the two hormones that we need to have? Prolactin and oxytocin. Prolactin again is responsible for production. Oxytocin is going to be responsible for ejection, milk let down, to contract the lactiferous sinuses for them to eject the milk out. Another function of the oxytocin, rather than enhancing the uterine contractions during labor and contraction of the lactiferous sinuses during lactation, it's going to be its role it plays in sexual arousal, orgasm in both males and females. Moving on to our second hormone that gets produced from the posterior pituitary gland, and this is going to be ADH, or anti-diuretic hormone. So from its name, what can I understand? I am a hormone. Anti means I am preventing. What's a diuretic? You have you ever seen a diuretic or heard about a diuretic before? Yeah, what is the function of diuretics? Who will be taking diuretics? Person with chronic heart failure. What else? Hypertension. Hypertension. What is the main function of diuretics is to, what's the term diuretic means? It's the release of water from the blood to the urine. So why do you want to reduce the blood volume? If you are getting the water to move from the blood to the urine, this means you are getting rid of the water in your body. Why would I need to do this for some of the patients? I have, like what you said, chronic heart failure. What's chronic heart failure? Patients with heart failure, this means what they do suffer from? Their heart fails to pump the blood or adequate amounts of blood to the tissues. So I want to have high or low pressure for me to pump the blood against. If I have weak heart, this is my chamber of the heart. I am pushing, I'm contracting this ventricle to pump the blood to move to my vessel in here. If I do have lots of blood in my vessels, would it be harder or easier for me to pump new blood in? If I have a lot of blood in my vessel, high pressure, will it be harder or easier for you to move the blood from the heart to the vessel? What do you think? Let's say on here. In order for anything to move, it needs to move from high pressure, right, to low pressure. The lowest pressure in your blood vessel is your diastolic blood pressure. Do you know around what number is your diastolic blood pressure? What do you think? Diastolic blood pressure. So systolic is 120 millimeter mercury, and the diastolic is lowest pressure in your arteries, 80 millimeter mercury. So let's say if I want to eject the blood here from the heart to the blood vessel has its lowest pressure, 80 millimeter mercury. So if I want to eject 1 liter of blood, let's say, the difference between the two pressures need to be 40 millimeter mercury if I'm ejecting 1 liter. All right? So what should be the pressure within my heart in order to pump this liter out? I need to have a pressure difference between the high pressure and the low pressure of 40 millimeter mercury. So what should be the pressure in my heart to pump this liter out if the difference needs to be 40 millimeter mercury? Do you understand my question first? Hello? All right, so if I want the difference between this pressure, this side, and this side to be 40, so X minus 80 equals 40. Can you answer this for me? Yeah, 120. So in order to pump this liter, I need to create a pressure of 120 within my heart here. I need to create 120. What if I have hypertension? Hypertension means high blood pressure. Where? In your arteries. So now the artery here doesn't have 80 millimeter mercury. Let's say it has 100 millimeter mercury. So I still need to pump this liter out. So I still need to have a pressure difference of how much? 40 millimeter mercury. So now X minus 100 equals to 40. What's going to be the pressure I need to create within my heart in order for me to pump this liter out? X minus 100 equals 40. So it's going to need to be 140. So in order to create 140 rather than 120, do I need to exert more or less effort? So in order to create this higher pressure, I need to have a greater effort performed or less effort performed? Greater effort. So if my heart is already failing, I have heart failure, is it something good to have high blood pressure? Or do you want to reduce this 80? Rather than being 80, I want to reduce it to be 70. For me to not create 120, but to reduce this 120 to make it 110. So what will I do in order for me to reduce the blood pressure in the heart failure patients? I need to reduce here the pressure within my arteries for it to become easier for my heart to pump the blood. So rather than having a pressure of 80, if it was normal and I have heart failure, I need to reduce it even below normal. I need to reduce it to 70. Why? in order for me to reduce the pressure that I need to create within my ventricle, rather than being 120, I need to reduce it to 110 for me to exert less effort. Why do you want the heart to exert less effort? Because it's failing. Does this make sense? Make sense? So why did we say all this? Just to know what is diuretic. What's a diuretic again? It's a drug that we use to move the water from the blood to the urine for us to reduce the pressure of the blood. Make sense? All right, so if I am a hormone called anti-diuretic, so what's gonna be the function of this hormone? To do what to your blood pressure? What do you think? I am anti-diuretic means what? Means I am preventing. What's diuresis? Diuresis means water loss in the urine. So rather than losing water in the urine, you're gonna prevent this from taking place. So what's gonna happen if you're not allowing this water to be released in the urine? It's gonna build up in your blood. And as you increase the blood volume, what's gonna happen to its weight? If you have two cans, one that has 10 liters of water and one that has 1 liter of water, and you put them on a table, which one applies greater pressure? The one with 10 liters or the one with 1 liter? 10 liters, right? So the greater the blood volume, the greater its pressure. So what's going to be the effect here of the anti-diuretic hormone that prevents the water loss in the urine? It retains the water, and as it retains the water, this is going to be increasing the blood volume. And as it increases the blood volume, when you increase the volume, the pressure of the blood gonna be increased. So what's gonna be the effect of anti-diuretic hormone on your blood pressure? It's gonna be increasing, again, increasing my blood pressure. It increases the blood pressure, all right? Does this make sense? Make sense? Another name for ADH, other name for anti-diuretic hormone is gonna be vasopressin. Vessel, pressin, I'm putting pressure. This tells you another function of ADH. It's gonna be constricting the blood vessels. So if you have a yard here, your backyard, and you have a water hose like this, and the water is not reaching all the way to this point. So what do you do to the water hose in order for you to get higher pressure of this water to reach this point in your yard? What would you do with this water hose? You're gonna be applying a pressure, exactly. You're gonna be compressing this water hose. And as you compress the water hose, what's gonna happen? You are reducing the diameter. And when I reduce the diameter, what's going to happen? This going to be increasing the resistance against the water flow. And as I increase the resistance, the water going to be traveling at a greater pressure. So what's the other name here of ADH, vasopressin? It's going to be vasopressin. And what do you understand from the other names? That it performs constriction of the blood vessels. In other words, it's going to be responsible to do what? To the pressure of the blood circulating. What happened to the pressure of the water here? Did I have an increase in the pressure of the water traveling out or a reduction in the pressure of water traveling out when I compress it? Increase or decrease in the pressure? I'm creating greater pressure. So it's the same concept. If I'm constricting the blood vessels, what do you think is going to happen to the pressure of the blood traveling inside? If you're compressing the vessels, pressure will increase. So two main ways by which you're going to be increasing the blood pressure under the effect of ADH. First one, you are retaining water, and as you retain water, this increases your blood volume. And as you increase the blood volume, the blood pressure is going to increase. The other way by which ADH is going to be increasing your blood pressure is going to be by performing constriction of your vessels. And as I constrict the blood vessels, this will be increasing the blood pressure as you are increasing the resistance against the blood flow. So two main mechanisms by which ADH is going to be inducing the blood pressure, increasing the blood pressure, and you can actually get them from the two different names. ADH, I'm preventing the loss of water out in the urine. Vasopressin, I'm performing constriction of the blood vessels. Questions? I think we won't be able to complete the chapter as I was planning to today. We're going to be completing this on Thursday. We're going to be completing the chapter on Thursday. Again, where can you find the most important diagrams that you need to prepare for this upcoming lab exam? Lab exam 4. You will see this lab exam 4 review on Canvas. So please make sure you are familiar with all those marked structures, all the marked structures on the brain and the eye. All right, those are the most important diagrams. Again, you need to be familiar with those diagrams as well, the fresh specimen diagrams. You might see them on the exam. All right. Questions? Anybody couldn't find this on Canvas? Yes, it should be available on Canvas. It's called the lab exam 4 between brackets review. It should be under the lab documents. So if you open here, So if you are looking at your course modules, we did remove all the weekly modules, and this is what should appear on here for you. So we've got the lecture PowerPoints, we've got the lab activities files. This is a module, and you will see on here under the lab activities files, you will see the lab exam full review. This is where you're gonna have those diagrams. Please make sure to download them. Please download them for the labeled markings to appear for you. For Lakeisha, I don't like to teach online. But if the campus will remain closed over the summer, my classes are gonna become online classes. Does this answer your question? So I don't usually teach online classes.